Hi everyone, in this video we're going to be looking at the liver. So what we're seeing here is a low magnification view of our slide, and so what you're seeing here is the edge of the liver, the outer edge. And so for example out here we have a bit of connective tissue visible, goes all the way around, you can see here and here, it's that connective tissue all the way around. So this is the Gleason's capsule, and as you can see this capsule goes into the interior, invaginates into the interior to subdivide the liver into lobes and lobules. And so you can see a bit of that here and here and here again. It's a bit of connective tissue just going into the interior. Okay, now this capsule can then be seen out here and going in deeper. You can see it following in. So all of this here is connective tissue, and then up here we see a portal triad. Okay, so what we're looking at here is a section through the liver. What we're looking at here is a hepatic lobule. So basically it's roughly hexagonal or pentagonal sort of structure, or at the corners of this structure you have these portal triads, what you're seeing up here for example. There's another one over here, there's another one over here, another one here, and all of this is around the outside of this structure, kind of defines the outer edges of it. And then in the middle, what you have is a central vein. So here's a central vein over here. Here's another lobule with a central vein right there. There's another central vein right there. There's another central vein, and again, we have these portal triads here, for example, around the outside of this structure. Now in this case here we have a fairly large amount of connective tissue, and these portal triads are fairly obvious, and that's because this is actually a slide of the pig liver, so it's a little bit easier to identify things here. The Gleason's capsule, as it goes in, has a bit more connective tissue. If we were to look at human liver, it probably wouldn't be quite this obvious. So let's take a closer look. Let's zoom in. So right now we're at lowest magnification, so we're at 40x. Let's zoom in and take a look at this 100x. And so again, right there at the pointer we have one of these portal triads. Let me try to find another one that's a little more obvious, or a little bit easier maybe. So there's another one over here, but I guess this is probably okay. We'll take a look at this one in a bit more detail. What I'd like to point out is that right there is a central vein, and I want you to notice that there's a bit of a radiating sort of appearance to the cells in this area here. So this is where you find the hepatocytes, and so they're arranged into hepatic plates, and they tend to be fairly long chains of cells that are all interconnected. And they're basically going from the edge, from where the portal triads are, to the central vein. And so if you look carefully, especially it's easier to see this at lower magnifications, the arrangement seems to be everything's kind of radially arranged around the outside of the central vein. So keep an eye out for that sort of thing when you're looking for whether you're looking at a central vein or not. Okay, so here's a central vein, and in this region we have a portal triad, which basically means that we have a hepatic artery and a portal vein, as well as a bile duct. There's a good chance that that right there is the bile duct. The bile duct would have a simple columnar or simple cuboidal epithelium, and the others would be typical blood vessels. So the artery would have a relatively small lumen, a simple squamous epithelium with a bit of smooth muscle around it. Again, an arteriole, and the vein or venule would just be more of a more collapsed lumen with a simple squamous endothelium. There might be some smooth muscle around it, but quite often there isn't. So let's take a look at this one and zoom in on it a little bit. So we're going to go to 400x. And so here we see this one I was saying earlier, it's a bile duct, and that is in fact a bile duct. There is a vein, you can see a collapsed lumen, not much going on around it. It's very difficult to really identify whether this is an arterial or not. We have relatively small lumen here, relatively small lumen here, relatively round, so it's very likely these are arteries. But the section doesn't really allow us to identify smooth muscle very easily. This right here, where are you? Right there. This could be a smooth muscle fiber, so it's very likely. Here's another example. So here we have an arterial, a bit of a longitudinal section through one, which is why the smooth muscle here looks like it's in cross section. Okay. There's a small venule right there, and again we see a simple cuboidal, probably simple cuboidal epithelium here. So this would be one of the bile ducts. Okay, so again we got portal triad here, and another one here. You can see some blood vessels. I don't really see. Well, there's a bile duct right here. Okay, so again, all these cells here beyond this are the hepatocytes. And in between the hepatic plates, what we have are the capillaries called sinusoids. These are much wider than a typical capillary. Let me see if I can try to increase the contrast here a little bit so we can maybe see them a bit more clearly. It's a little bit easier with better stained slides, but this one has very faint staining, unfortunately. Okay, so this would be one of the hepatic plates. And then this region right here looks kind of empty. That would be one of those sinusoids. And this all leads into the central vein here. Okay, so again, here's another sinusoid. Here. And another one, kind of a more of a cross section through one over here as well. Okay, so we have some sinusoids visible here. Let me zoom out, see if I can see something a bit more clearly at low magnification that I can show you. It's a little bit more obvious. Okay, might be able to see something a bit more clear in this region. Let's zoom in on this. Okay, so here we have some of these sinusoids are a bit more obvious. So here's one. Here's another that's a bit more highly branched here. And again, it's all flowing in here into this central vein. And again, here's a sinusoid. Here's another pretty obvious sinusoid. And again, lots more visible, especially in this area. And again, this cluster, long chain of cells would be one of the hepatic plates. So again, hepatocytes connected to one another. Now, with this staining and this magnification, you definitely will not be able to see the space of Dissay. And you will not be able to see the Kupffer cells here because they're not stained for in this case, and they're just macrophages. So again, it will be a difficult thing to try to identify under the microscope in this situation. So really the only thing that you can identify here with any certainty would be the hepatocytes as well as the sinusoids, which are, in this case at least, relatively obvious. Okay, so again, main structures or main things to notice: hepatocytes, sinusoids, central vein, and then again, here is an arterial or a small artery, very small lumen, fairly thick amount of muscle here, There is a bile duct. The epithelium here is actually starting to look a little bit more columnar, simple columnar. Here's a smaller bile duct over here, which is more simple cuboidal. And again, this is likely to be a vein over here, lumen of a vein. There's a smaller vein over here. And again, the arteries and veins that are found within the portal triad will branch off and run along the length of the connective tissue that separates the different lobules from one another, and so you have blood flowing not just from the portal triad but from also the areas around the portal triad, around the outside where the capsule has moved around and outlined the outsides of these lobules. Okay, so hopefully this has been informative, and we'll see you in the next video.